Good morning, everybody. I'm starting out today with my mask on. I hope everybody's wearing their mask, but since I'm in my office alone, I'm going to now take my mask off, make it a little easier. Anyway, it's a pleasure to welcome everybody to kick off this year's annual Pioneers in Biomedical Research Seminar Series for the Fralin Biomedical Research Institute. We're very glad everybody's here or somewhere. At least we can see you online, and we're looking forward to a great series this year. So just by way of quick background, the way we do these seminars, as you know, we have several centers here at the Institute, and each center or focus area got to select several speakers for their part of the program this year. So our very first speaker is jointly sponsored by the Fralin Biomedical Research Institute, but also by our Center for Transformative Research on Health Behaviors, led by Dr. Bickle and now Dr. Stein and Dr. DeFelice-Antonio as well. And so it's my pleasure to welcome Dr. Warren Bickle to introduce today's speaker. Warren?
Thanks, Mike. So I am very pleased to introduce our first PBR speaker, Ashley Gearhart. Dr. Gearhart is Associate Professor in the Department of Psychology at the University of Michigan. She received her bachelor degree from the University of Michigan and her Ph.D. from Yale University. Ashley is also a licensed clinical psychologist. She has made significant contribution and is recognized as a world leader in understanding that food can function as an addictive substance. Now, the DSM, the Diagnostic and Statistical Manual of the American Psychiatric Association, does not recognize food as an addiction, and thus this is a controversial area. Indeed, some question how we can be addicted to something that we need to consume in order to survive. However, the large increase in eating disorders such as obesity provides affirmative evidence for considering such a concept. One of Ashley's seminal contributions was the development of the Yale Food Addiction Scale. The food scale identified certain foods more likely to trigger dependence symptoms, such as high fat and high sugar processed foods. In 2014, she published a meta-analysis of the Yale Food Addiction Scale that suggested among relatively large samples, approximately 20% of individuals met criteria for food addiction. Her interests in the topic are broad. For example, she addressed the policy implications of food addiction. She's modified this scale to a briefer form, created a version for children, and has been instrumental in translating it into several languages. But that's not all she does. She does a wide variety of other types of research. The ones that caught my eye in particular was she's examined the substitution of alcohol for food during weight loss, the role of poverty and stress on eating, and the neural response to food marketing as predictors of food addiction and weight gain. She's also has one of the most unique laboratories. Her laboratory simulates a fast food burger joint, and that way she's able to examine specific contextual features that engender food choice and eating. She's been a highly productive researcher with over 130 publications. She has received several awards, including the Association for Psychological Science Rising Star, and she is a regular recipient of NIH research grants. And for those who have not met her, she's truly a delight. Just one notice for everybody, there's a questions tab at the bottom, so if you have questions, please answer them there. And without further ado, let me introduce to you Ashley Gearhart.
Now I'm unmuted. Hello, everyone. Brad, I'm having a hard time starting my video. It says I'm unable to do so at the moment. Would you be able to start my video? It says it's blocked by the host.
Brad, can you do that for Dr. Gearhart?
I'll give it another second here, but the other possibility is you may not be able to see my smiling face, and I can just share my slides with you and audio do that. But if Brad does come on, I will. Oh, there we go. There's my video. Hi, everyone. Nice to see you virtually. I wish that I could be there with all of you in Virginia Tech. I am just incredibly blown away by the high quality of work that happens at your institute, and wish I could have been there in person to kind of break bread and hear more about the wonderful research that you're doing. But I've had already some wonderful and stimulating conversations, and I'm really looking forward to sharing with you my research today. So I'm going to get started here and share my screen with all of you. One quick second here. All right, and we'll get going. All right. Hopefully you can see this screen. Please do speak up if you're unable to see a delicious looking glob of chocolate ice cream. This is actually a advertisement that recently came into my email from a ice cream store in Ann Arbor that's absolutely delightful, that I really enjoy. And this is their Chocoholic ice cream. So, you know, we hear a lot of addiction language, craving, chocoholic, carb addict, these sorts of things that are actually even used in the marketing of really rewarding foods. And part of my scientific endeavor, as Dr. Bickle discussed in my very kind introduction, is to scientifically really probe, where does the line exist between things that we think of as traditionally addictive and the foods that are now dominating our food environment? And so I often like to start my talks by taking a little bit of a historical perspective to think about, how do we understand and define as a field, as a society, what is addictive? And it's a field that has been wrought with controversy and debate. And so this is some cocaine. And what we have seen is that, you know, for many, many decades, there was actually a lot of debate about whether cocaine could be addictive, especially the 60s and 70s. The idea was that addiction was driven by physical withdrawal, the experience of shaking and vomiting and illness that you see so clearly with things like alcohol and with opioids. And cocaine has a much more mild physical withdrawal syndrome. And so,
Dr. Gearhart, can you
yes.
go into full screen mode, please? We can see the
Let's
other
see.
slides here.
Okay, you, for some reason, it is sharing the screen that is on the other side. So I actually am in full screen mode, but it is showing it on one side, but not another. Let's take a look. Let me try this. So when I'm starting this, Brad, from the beginning, I just hit start and I'm now able to see the slideshow in full screen on my side on a screen. But
Yeah,
you
so
guys,
go ahead and stop sharing, and then
okay.
you may see a second screen there that you can share. It should say screen 2 maybe.
Okay, let's give that a shot. There we go. All right, share. Excellent.
We see the extended. Yep, that's perfect.
Okay, excellent. Thank you for bearing with me with this new technology of sharing these guys on a new setup. So I'll just jump right back to where I was and we'll get going. But the idea was that there was a lot of controversy around the idea that cocaine could be addictive because it didn't have that traditional physical withdrawal syndrome of illness and shaking and potential death that we saw with things like alcohol and opioids. So there was a lot of hot debate about whether we could consider it was addictive, despite the fact that there were high cravings and a lot of suffering and a lot of loss of control with consumption. But as we moved away from physical withdrawal as kind of the core feature that defined how and why people got addicted, you know, it became added into the DSM and DSM-3. But then in our next debate was really around cigarettes. And so again, now we have a substance that people crave, they use to their detriment, they can't stop even though they want to, but in many ways it didn't look like our other traditionally addictive substances. There wasn't an intoxication syndrome. It was legal, it was cheap, it was easily accessible. But as we know, it's now, you know, the #1 leading cause of preventable death around the globe. And there's generally been scientific consensus that despite its lack of an intoxication syndrome and its easy accessibility, that's a highly, highly addictive substance. More recently, in the 2000s, we've had debates about gambling and whether behaviors that are getting progressively amped up to be more and more rewarding through often our technological advancements, whether you're able to develop addictive processes to something you're engaging in where you're not even ingesting a substance into your body per se, but can just the experience of engaging in something that's been designed and heightened and optimized through technology to really hook certain people, even though you're not ingesting a substance, is that effective enough at triggering addictive compulsive behaviors? And in the current most recent iteration of the DSM-5, the gambling has now been included as an addictive disorder, with internet gaming disorder being as a potential kind of waiting in the wings of another addictive disorder that's in the appendix. So as you can see, kind of the take home point from this is the idea that our understanding of what is addictive and what should be considered addictive has been rapidly evolving, even in the last few decades, to understand what we should classify as something that can trigger addictive responses. And so I like to think about that in the context of a rapidly changing and rapidly evolving food environment. What you see on the screen are some bananas, which are naturally high in sugar, and some nuts, which are naturally high in fat and are pretty energy dense. And we know that as humans, we are evolved to make sure that we are getting sufficient calories. And there is so many overlays of neurocircuitry and biological and psychological systems that make sure that we are attending to high calorie, high rewarding ingredients like these. Because for much of human existence, these ingredients were extraordinarily scarce, and we were often faced with famine. But as we have done in so many other domains, we have moved through technological advances where we are now so incredibly effective at stripping out the most rewarding ingredients of sugar and other carbohydrates and fats into their purest forms, and then combining them into thousands and thousands of unique and novel and constantly changing food combinations that are so different than anything our brains really evolved to handle. And now these ultra processed foods that we've designed and created through human ingenuity are the dominant source of calories in the modern food environment. And as we see, as food environments start to become dominated by these foods, you see what rises with it are high levels of obesity, diet-related disease, and binge eating, loss of control eating. And now it's thought that dietary contributors related to the consumption of these ultra processed but minimally nutritious foods is now the #2 leading cause of preventable death in the United States and in other Western countries. So it really is a changing food environment that we're all kind of the current guinea pigs. We don't really understand exactly what these sorts of food items and food types do to our psychology, to our biology, but we're certainly seeing that they're having negative consequences that occur even though people really want to have control of their eating behavior and manage their weight effectively. People are repeatedly failing to do so. And so this really brought up the question for me when I was starting graduate school about the idea of, have these certain foods cross the line from something that's just yummy and tasty and pleasurable to something, for at least for some people, are powerful enough that it's capable of triggering an addictive process. So the next thing I want to jump into is kind of the first step that I took to try and probe this question scientifically. And at the time I started entering into this work, I was noticing that there was more and more interest in this overlap between addictive disorders and problematic eating behaviors, led in large part by beautiful, elegant animal models, and also Norvolcal and NIDA. But some of the things that I noticed is that people were using things like obesity as a proxy for an addictive phenotype. And as we know, obesity is an incredibly heterogeneous condition that can result from genetics, from medication side effects, from physical inactivity. And so that felt like almost using lung cancer as a proxy for an addiction to nicotine, not quite specific enough to what we're really diving into with addiction. Alternatively, people were using things like self-identifying as a chocoholic or a carb craver, but who really knows what that means in the context of a society where we use addiction language a lot to describe pretty much everything under the sun. And so I really wanted to develop a psychologically and psychometrically rigorous tool to be able to operationalize the individuals who might be showing an addictive response to these sorts of foods. And I decided to use the diagnostic criteria of the DSM-4 at the time I developed the original version, and now I've updated to DSM-5 for substance use disorders. And why I wanted to do this is that I wanted to kind of take the same questioning process that I would take if someone came into my clinical office and I was trying to distinguish whether they were a social drinker, actually had an alcohol use disorder, an addiction to alcohol, and using those same criteria that we would use in the context of alcohol, opioids, cigarettes, et cetera. It seemed like a really important place to start to ensure that we were talking about the same construct. And so I created this measure that has 35 items with frequency response options about how frequently they're endorsed, that provides both a symptom count and a diagnostic option for scoring. And it discusses symptoms like loss of control, craving, continued use despite negative consequences, tolerance, and withdrawal, those core characteristics that we use to define addictive disorders. This measure at this point has been evaluated very widely for its psychometric properties and non-clinical samples, clinical samples of individuals with obesity, eating disorders, bariatric surgery patients, and also large epidemiological samples like the Harvard Nurses Health Study. And what we see is that it has strong psychometric properties of internal consistency, test-retest reliability, construct validity, discriminant validity, and incremental validity. And so I could kind of rest in the idea that this is a scale that is at least providing us with a good, more rigorous tool to be able to start to further scientifically evaluate the question of whether addictive like eating exists and for whom. And speaking to kind of, I think, the global concerns when it comes to kind of the broadening issues with our diet and with our current food environment, the measure has been translated and validated into over 10 different languages for both adults and children, and has been cited over 1,000 times. So I wanted to give you a little bit of a flavor of what the sort of questions that I'm asking look like. So here's a few. I had such intense cravings for certain foods that I felt like I had to eat them right away. I needed to eat more and more to get the feelings I wanted from eating. And I really wanted to cut down on or stop eating certain kinds of foods, but I just couldn't. And to give you a sense is that what we do in this scale as well is that we prime people to specifically think about those types of ultra processed foods that are engineered to be high in refined carbohydrates like sugar and fat, and to think about specifically those types of food when they're filling out this questionnaire. So, this YFAST scale exists. What does this potentially tell us? When we look at this behavioral phenotype, you can see here endorsement of different types of food addiction. There's mild, moderate, and severe based on the current DSM-5 thresholds, and then total food addiction. And you can see how this differs by weight status. And so what you can see if you look here at total food addiction prevalence in the validation article for the most recent version of the YFAST is that we see that about 33% of individuals with obesity meet the threshold for food addiction. And this is based off of the same thresholding that we use with any other substance use disorder in the DSM-5, 2 or more symptoms plus clinically significant impairment or distress. So we do see here that not all individuals with obesity meet for food addiction based on the YFAST. And we also see that when you look at the normal weight and overweight categories, that there are people who are endorsing this behavioral phenotype that aren't necessarily having elevated body mass index. When we've kind of dived into that a little bit in my lab to try and figure out what's going on, is that what we generally see for those groups is either they're quite young, and so their propensity to overconsume these foods has not quite shown up yet in their body mass index, or they're using compensatory behaviors like extreme dieting, fasting, or purging that might not reflect the actual compulsive relationship with food and their body mass index. Overall, when we look at meta-analyses, we see that about 15 to 20% of individuals meet the food addiction diagnostic threshold. And this is very, very similar to what we see for the percentage thresholds for other legal and easily accessible addictive substances like alcohol and cigarettes are about at the same prevalence rate of about somewhere between 15 to 20%. So what might this tell us? So we're seeing that there's this behavioral pheno subtype that seems to be an endorsement of addictive-like indicators in the consumption of these highly rewarding foods. But does this tell us anything that might be of relevance to kind of understanding and treating problematic diet-related disease or obesity? And so this study recently came out from a group in Stanford. It's the Diet Fits trial that has 609 adults, pretty diverse of a sample. And the goal of the sample is really to do a randomized trial of a 12-month low fat or 12-month low carbohydrate diet for weight loss. And one of the secondary aims of this study, kind of pitting what's better, low fat or low carb, kind of spoiler alert, they found that in general, both worked pretty equally effectively. One of their secondary aims was to try and understand what were baseline psychosocial and demographic factors that might predict people who did not respond well to these treatments. And in this study, they included a whole wide range of about 35 different predictors that they then pitted against each other in stepwise logistic regressions to identify what would predict study attrition, which was about 1/4 of the participants, and what predicted among trial completers the tendency to gain weight rather than lose weight over the course of this 12-month trial. So what you can see here are the factors that predicted a tendency to drop out of treatment. And what I really want to draw your eye to is that food addiction was found, even accounting for all these other variables, as the highest predictor of a greater likelihood of these individuals to drop out of treatment, with 43% being more like, with a greater prevalence of 43% likelihood of dropping out. When it comes to trying to predict weight gain at 12 months, again, we see that food addiction is the highest predictor of individuals who are prone to gain weight rather than lose weight in these weight loss trials. And so in seeing this work that I wasn't involved in, what I took home is that this phenotype that we've kind of zoned in on may be particularly important for telling us about individuals who are at heightened risk of failing when it comes to responding to our traditional treatments that are about focusing on, changing your diet quality and reducing your calorie intake. And that really makes sense if we think of part of what's going on is there's an addictive pull to the ultra processed foods that are so prevalent in our environment, that just helping people try and change their diet without additional support may not be enough for these individuals, and we might need some specialized treatment to help them be more successful. So my next idea is so what might that be? What might be going on for these individuals? Can we start to dive in to different mechanisms that might be at play that we might want to target in our treatments? And so one of my major areas of focus is in the realm of reward and sensitivity to cues and the ability for it to drive wanting and desire for these sorts of foods. And then also what happens when individuals are actually consuming these highly rewarding foods. And so early in my career, after we created the YFAS, I collaborated with a group at Oregon Research Institute with Eric Stice and Sonia Yoakum, and we wanted to look at what were the reward-related neural correlates of the Yale Food Addiction Scale. And so this is a sample of 48 healthy young women. They didn't currently have, we were screening out for eating disorders as part of a separate trial. And what we found is that overall, we had a body mass index that was on average in the overweight range, but ranged from normal to obese. And in our analyses, we excluded for all Axis 1 diagnoses, including eating disorders, and we controlled for body mass index. So we were really looking at addictive-like eating outside of the context of both eating disorders and looking at it independent of body mass index. In this setup, which the Stice lab uses, and that Dana Small at Yale was kind of one of the early people to pioneer to look at differences in neural response to anticipatory and consumatory food reward, individuals in the scanner would see either a picture of a chocolate milkshake or a glass of water. That would predict consumption and delivery of a small amount of either chocolate milkshake in the scanner or a tasteless solution. And why we used a tasteless solution rather than water is actually effective at activating the gustatory cortex in the brain. And this tasteless solution kind of mimics saliva. And so it doesn't actually activate this sense of taste in the brain. So it's kind of a more precise control stimulus. So we did a number of trials where individuals were having this anticipation of either getting chocolate milkshake or this tasteless solution, which we labeled as water, and then actually getting chocolate milkshake or this tasteless solution. So we could look at the anticipation and the consumption of the substance. And so what did we find? So we found that the YFAS in this scale, that the higher the individual's food addiction scores were, the greater reward-related neural activation they had in regions such as the anterior cingulate cortex, the medial orbital frontal cortex, and the amygdala when they were viewing the milkshake picture relative to the water solution. So when they're anticipating getting this rewarding response, when they're seeing that cue, we're seeing greater activation in these brain regions when looking at the YFAS continuously. We found that overall these brain regions, when we kind of think about what are some of the mechanistic features that are thought to be associated with these brain regions, is that in the context of substance use disorders, they're often implicated in things like craving, enhanced motivation, and the greater incentive salience of cues. We then split our groups into kind of a high and low food addiction group to directly compare these extreme groups. And what we found is that individuals in the high YFAS group relative to the low YFAS group, when looking at the milkshake cue relative to the tasteless cue, showed greater activation of the caudate. Interestingly, this difference in neural response wasn't driven by differences in greater activation to the milkshake cue, but rather a diminished response to the tasteless cue in the high food addiction group relative to the low food addiction group. And to me, this is interesting in this idea of kind of hedonic adaptation that we see in the context of substance use disorders, is that over time, individuals can start to, their reward system kind of only gets out of bed and responds to the addictive substance that they're interested in. Things that aren't the addictive substance, like social rewards or physical activity, start to become less and less effective at engaging reward systems. And so there's really seems to be this contrasting effect between the highly addictive rewarding substance that can still activate the reward system and other features and other rewards in the environment that are ineffective at activating the reward system. We then looked at the actual consumption of the milkshake, and here we saw far fewer effects. Overall, the only effect that we really saw during the consumption of the milkshake was a reduction in the lateral orbital frontal cortex when individuals were consuming both the milkshake and the tasteless solution relative to the low food addiction group. And what the lateral OFC has been implicated in part, has been inhibitory control, the ability to kind of put on the brakes. And so what we're thinking about from this kind of early neuroimaging study was this potential idea that individuals who are endorsing more symptoms of addictive-like eating might be particularly prone to experiencing motivational drive and heightened reward response to cues that signal the availability of an addictive substance here, of a chocolate milkshake. And then when they're consuming the chocolate milkshake, the brakes that might help them inhibit the amount that they're consuming is diminished. So this is an early study. It's a group of individuals that overall were not extremely highly clinically relevant because we were excluding a lot of individuals with severe disordered eating behavior, but it is a very clean sample, and that we're controlling for body mass index in all of these analyses, and none of these individuals met for an existing eating disorder. So this really makes me think about one of the mechanisms that we might be thinking about and treating and attending to more for folks that are endorsing addictive-like eating is this heightened reward-related response to cues and this stronger reward drive. And coupled with that is that lateral OFC suggests that there may be something going on in the inhibitory system. And this really makes me think of Dr. Bickel's theory of reinforcement pathology, as that a combination of a high reward drive for something coupled with a deficiency and inhibitory control systems is a particularly toxic combination, and having people kind of compulsively overconsume rewarding substances. And so here, this is a group of of researchers, again, this isn't work that I was involved in, but with another group of James McKillop's groups in Canada, looking at individuals who had high versus low food addiction and finding that individuals with higher levels of clinically significant food addiction did show greater delayed discounting, a tendency to discount future rewards more than immediate reinforcers. And then when looking at predictors of what were the best predictors, including demographics and other measures of inhibitory control difficulties, what this group found is that the best predictors of food addiction were higher levels of urgency, a tendency to act impulsively when one is in either a high level of negative or positive affect, as well as a tendency to have higher levels of delayed discounting. And what was interesting is that in this sample, they did not find these same results when it came to looking at obesity on its own, but it really seemed to be driven by this food addiction phenotype that was experiencing these differences in delayed discounting and heightened urgency. And so again, thinking clinically, in addition to wanting to target those reward systems and trying to equip people on how best to handle cues and craving and desire, it also suggests that we need some additional scaffolding with things like potentially future episodic thinking or greater enrichment of other non-addictive rewards to help individuals respond to inhibitory systems that may not be benefiting them in a food environment that is just completely dominated by highly rewarding, highly enticing food cues if they are showing this addictive-like phenotype in their eating behavior. And so I think of these mechanisms as being extremely key to what we're targeting, and also the key mechanisms that are at the heart of other addictive disorders as well. So this is consistent with the idea of conceptualizing at least this subphenotype as potentially showing mechanistic differences that are implicated in other addictive disorders. But I also never want to lose sight of the fact that the stimulus that is the focus of the addiction is really, really key. And so when we think of this in the context of other addictions, I mean, we're currently living this, right, with the opioid epidemic, where when we had technological advancements, where we developed through pharmaceutical companies opioids that were more rapid acting and more accessible, we saw all of a sudden individuals who maybe had heightened tendencies of reward or emotion dysregulation or inhibitory control, and then had greater access to these addictive opioids when they were told they were safe. That is a public health that's a recipe for a public health crisis. And so when I think about what's going on in our food environment, is in many ways we've done the same thing to our food environment that alters the addictiveness of a drug. And so when we think of what makes a drug of abuse highly addictive, it's when we increase the dose of the addictive agent. So when we go from something like a low alcohol beer to something like a shot of whiskey, there's a higher dose of the addictive agent in it. But also extraordinarily important is a rapid rate of absorption. We can take something that does have an addictive potential, like a stimulant, and we can, if it has slow release, use it as a treatment in the context of something like ADHD medication. But if it becomes rapidly absorbed or smoked or snorted and is able to hit the brain extraordinarily rapidly and often very briefly with a crash, that rapid rate of absorption might even be more important in predicting the addictive potential of a drug of abuse. So when I think about these highly processed, ultra-processed foods in our environment, we have been increasing the dose of rewarding ingredients like sugar and fat that naturally do not come occurring at very high levels. And we're increasing their rate of absorption by stripping out things like protein and fiber and water that allows especially the carbohydrates to hit the system at a rate that is much faster than we see in minimally processed naturally existing foods. In addition to this, we're seeing these unique combinations. Like when you think of naturally occurring foods like fruits or nuts, they either seem to come high in sugar, like in the context of that banana that I showed you earlier, but not high in fat, or they're high in fat, but not in sugar, like in the context of the nuts. Kind of the only combination that we see in nature are things like breast milk or coconut milk, which are pretty hard to come by, and kind of, you're not expected to be consuming breast milk for the majority of your life. And so the fact that the majority of these foods that we're interacting with in our current modern food environments have high doses of both highly rewarding carbohydrates like sugar and fat, plus sodium and flavor enhancers and all sorts of other ingredients, and a rapid rate of absorption, to me strikes me as having very high parallels that makes me think that these foods have maybe moved along the path as something that was rewarding and beneficial to something that has a high addictive potential for at least some individuals. And so that has been one of the goals of my lab. And Erica Schulte, who's a grad student of mine who's now at Penn as a postdoc, has really been diving into. And I have to say, like, Alex is doing that, who is now on your faculty here. Alex, I feel like I'm going to butcher your last name. I realized I wanted to check it with you today, but I didn't. Alex Di Feliciano. I'm just going to like go with that. Alex is doing some phenomenal work, which is just completely so beneficial and foundational to the work that we're doing in my lab, of really getting deeply into the physiology and both the gut and in the brain about how are these potential foods and ingredients kind of going up the system and really activating reward related circuitry in a way that maybe mimics drugs of abuse. And so this is a table from her work kind of showing that fat and carbohydrates might work through different pathways to both kind of simultaneously end up activating striatal dopamine in a way that is quite heightened, in a way that would mimic what we think of as drugs of abuse, being able to kind of artificially activate striatal dopamine in a way that goes beyond what people are kind of designed to handle. So her work is just absolutely foundational, and I'm so excited to continue to see more work coming out of her lab. So my lab really focuses on the behavioral side of things. And so when we were doing some work, when I started this, we called it food addiction for the lack of a better term, of being able to kind of hone in on what types of foods might be capable of triggering this addictive process. But it's very clear that not all foods are equally likely to trigger this addictive-like phenotype. And so we decided to do a study to say, well, what foods behaviorally are most likely to have people endorse the behavioral phenotypes of addiction that we measure in the YFAS. And so we took out all priming of what foods we wanted individuals to think about. We had them fill out the YFAS and then complete a serial ranking task about which food were they more likely to experience this with: chocolate or apples, French fries or cookies. And what you're seeing here is the result of our forced choice ranking task. And so these were the top 10 foods that were ranked in our nutritionally diverse sample of 35 different foods as those that were most likely to trigger addictive-like eating behavior based on the YFAS. And so there's no surprises here, right? Chocolate, ice cream, French fry, pizza. I think we can all resonate with that. And these are all foods that are highly processed and that have high doses of foods, of ingredients like refined carbohydrates and fats, plus often salt and other flavor enhancers. What foods are not likely were people reporting that they weren't consuming in an addictive way? All minimally processed foods that exist in nature. Foods like apples, brown rice, cucumbers. I always enjoy that beans are the very last food. I feel like there might be like an aversive property to like consuming too many beans or broccoli. And so, what are we seeing here? It's that these nutritious foods that were the majority of our food environment for the vast aspect of human history are not foods that people are prone to consume in this addictive way, but foods that we have designed and altered to be highly rewarding and highly processed are the sorts of foods. And so I've been kind of refining the term, and in my lab, we've been referring to it more as these highly processed food addiction to kind of get specific that it isn't just the act of eating any food, but these certain foods that seem to interact with individual risk factors that then trigger this compulsive eating behavior. When we tried to kind of dive a little bit deeper and a larger community sample to use hierarchical level modeling to understand for whom are certain attributes of these foods maybe more or less risky, what we found when we were looking at processing, just was it a processed food or not, fat and glycemic load as a marker of kind of rapidity of absorption of carbohydrates and its ability to spike blood glucose, what we found is that individuals who had more addictive-like eating experienced overall more difficulty with processed foods. The more addictive eating they had, the more processed foods were particularly likely to engage them. And that it really seemed to be driven more by these foods rapidly absorbed carbohydrates that had high glycemic load. and that fat was kind of this additive factor, but once accounting for glycemic load, it didn't seem to kind of add much more to the story. So in my lab, this has kind of got us kind of focusing a lot on these rapidly absorbed carbohydrates as maybe the key active ingredient in these addictive foods. But then I think of fat as often this kind of supplementary factor that's hitting an alternative pathway to go up to the dopamine systems in the brain. And so when you have rapid refined carbohydrates plus fat, you get a 1-2 punch. And so I think, some of the factors that we're thinking about, again, about, well, when we're trying to understand, is that sufficient to know that something's addictive? There's still so many domains to look at that are so fascinating. I mean, this is a table from DiChiara's group trying to understand kind of different substances out there and what may or may not be addictive. And here they're talking about sugar some and kind of going through, what has this neurobiological effect of dopamine release in the striatum and then the behavioral correlates. And then looking at some clinical symptoms of things like craving and impaired control, which are clearly there in the context of sugar, and that it can also have use despite harmful consequences. Tolerance and withdrawal have not been studied deeply in the context of the foods in our environment. And so you'll see that those are kind of some questions that are hanging out there when considering overall addictive property. And they also have some really important clinical predictors, and that we know that an experience of withdrawal when people are trying to cut down on their addictive substance can predict those who are most likely to relapse and be a clinical target. And so we hear anecdotally all the time in our lab that people will say, oh yeah, when I cut down on carbs, when I cut down on sugar, I experienced headaches and irritability and craving, and much of the withdrawal sort of syndrome that you might expect to hear was something like cigarettes, but there were no tools and no strategies out there to really focus on that. So again, Erica has collaborated with me, and we've created the highly processed food withdrawal scale to create a scientifically validated measure to start to try and evaluate whether individuals are endorsing signs of withdrawal over the course of trying to get off these highly processed foods, which are dominating our food environment. When people try and switch to a healthy food environment, do they get this rebound withdrawal type effect? And so in this study of participants, 231, about half female, who reported cutting down on these foods in the past year, we created this measure by taking drug withdrawal scales and adapting them and focusing on factors like negative affect, craving, fatigue, and headaches. And what we found in this scale is that individuals who endorsed higher aspects of withdrawal when they tried to cut down on these foods were more likely to experience addictive like eating based on the YFAS, also more likely to have higher BMIs, and they were people who had a higher history of weight cycling, of going from gaining weight and losing weight and repeatedly failing to maintain their weight loss successes, so kind of a high relapse phenotype. And that this highly processed food withdrawal scale predicted failure in the last diet attempt above and beyond both addictive like eating and body mass index, suggesting that it was kind of having this additive effect to trying to understand who might be struggling to maintain healthy dietary change. And one from a construct validity perspective, we were really interested in the timeline in which these symptoms kind of peaked and then may go down. And what we see with other addictive disorders is that really the first 5 days seem to be the period in which the symptoms are most intense, and then they slowly start to taper off over time. And usually you see like a pretty big drop at about 6 weeks to 2 months out. And so when we looked at the time course of this with individuals, we saw the same time course where they said it was really kind of days 2 to 3, 4 to 5, where these symptoms were the worst, and then they got better. And so when I think of this again from like a clinical perspective, what might this mean is that right now we often say, okay, here's your new diet, cut out these, try and eat less fat, try and eat less of these sorts of carbs, and we'll check in with you in a couple of weeks. And what this work is suggesting that at least for an addictive phenotype, those first week might be a period that's really marked by emotion dysregulation and heightened craving craving and cue responsivity that we might need to target treatments around to help people stay in treatment and be successful. We were just getting ready to do a tightly controlled experimental design using neuroimaging and psychophysiology and ecological momentary assessment with like experimental diets that were calorically similar but differed in the rewarding quality of the foods, and to do it prospectively to look at withdrawal when COVID hit. So that is currently on pause, but we're trying to do it virtually right now so we can further probe into, is this just a retrospective recall bias, or are we actually seeing biological and psychological indicators of withdrawal when individuals are trying to cut down and cut off of these highly processed foods? I'm being really mindful of time. I'm realizing that there's always more to talk about than I think I'm going to get to. And so I have like a kind of natural pause here, and I was going to pivot into some work I have on kids, but I both wanted to check in with my moderators to see if you'd like me to take a moment and answer questions here, and how we're doing time-wise, what you'd like me to prioritize.
Yeah, so we have about 15 more minutes. We have several comments or questions. And very quickly, could you tell us about the problems with BMI, and are there other measures that are more specific that would better be, physical indicators of food addictions, perhaps body fat percentage? And is it possible to distinguish the cognitive aspects of food ingestion versus the neurometabolic aspects?
Those are great questions. I'd be happy to talk about that. So I do think, body mass index, as we know, is a tool that has a lot of error with it. It's height and weight, but it doesn't break down into things like actual body fat mass or bone mass or muscle mass. And so your body fat percentage and other measures that kind of get more precise to adiposity are perhaps more effective. But I think overall, trying to capture a addictive phenotype by looking at the impact on the body is always going to be problematic. And so again, I just like to use this example of, let's say, cirrhosis of the liver, right? We know that individuals who are excessively consuming alcohol are at much heightened risk of cirrhosis of the liver. But we also know that the vast majority of individuals, even individuals who consume alcohol excessively, don't go on to have cirrhosis of the liver. And certain genetic factors and the way they metabolize alcohol can result in whether this biological process occurs or not. And that also many people can have cirrhosis of the liver without necessarily having an alcohol addiction. There might be other genetic factors or even excess sugar consumption that can lead to the cirrhosis of the liver. And so I really like to, in the context of addiction, think of those biological bodily outcomes as really key and important to why we care about this from a public health perspective, but that there's not a one-to-one relationship, and people's relationship with food and the way that it actually represents in their body and their body type. I think of this a lot when I think of the teenagers in my studies and the young adults who are clearly showing an addictive phenotype often with food, but they're so young and their metabolisms are so high, and they're not at risk at this moment in time for necessarily severe obesity or the associated factors of type 2 diabetes or hypertension. But if this established pattern continues over time, they're going to be at heightened risk as it interacts with their own background. Thinking about cognitive factors and biological factors, can you disassociate them? I would say that this is one of the trickiest parts of doing food research, because I've done alcohol research. That was my original training. And we would have these beautiful placebos, and we could really kind of disentangle what was going on with expectancy effects and cue and context effects, and disassociate that from the actual biological process of ingesting ethanol. With food, I haven't been able to find a great placebo control, because the taste so obviously kind of tells you whether you're getting sugar or not, or fat or not per se. And that there's a lot of controversy over artificial sweeteners, and that they're not an active placebo. Like, they certainly have effects on our brain and on the metabolistic system. And so I think that it's really tricky to disentangle those purely. We are trying to do work in my lab by really looking at expectancy effects and seeing kind of how people expect different experiences and the ability to experience pleasure or reduce negative affect related to different food attributes, and then trying to map that onto what happens when they really do eat that food. But it's really hard to completely blind them. So it's not a pure test. If anybody has any ideas, I'd love to hear it. Would you like me to continue, Warren, with some slides, or would you like me to take
Continue.
some more questions?
I think that's, you've answered our questions at the moment. Please continue.
Great. And I'll check in again and make sure, like, I'll give you about 5 more minutes of science. I'll try to prioritize a couple things, and then I'll make sure I leave 5 minutes at the end to address any other questions that have come up. So I like to think as well about the uniqueness of food in the context of development. When we think of things like alcohol or marijuana or cigarettes, early exposures are happening often in adolescence or early adulthood. In contrast, if we saw an ad like this, where it's like, we have the youngest customers in the business. If your baby's having a hard time, you're drinking your breast milk, add a little Heineken to it, we'd all be kind of appalled. But this is actually an old 7-Up ad. And we are seeing that infants, preschoolers, in utero exposure to high levels of sugars and other ingredients that we think of as potentially paralleling addictive substances are occurring so early in development. And so even if these foods and substances have less addictive potential than something like a beer, the chronic and persistent early exposure, when the brain is so malleable and early preferences and kind of neuroadaptations are occurring, is really concerning. And so I've been looking to investigate this in children. So we've created a YFAST for children, and in a sample of 8 to 12-year-olds from just the surrounding community in New Haven, we found that 7.1% met the food addiction criteria on the YFAST, and on average, some children were endorsing 2 symptoms of addictive-like eating. And what we found is that for those children, that the children who had more addictive-like eating, they had elevated body mass index percentiles, they had more overeating that was driven by emotions, they had less satiety responsiveness, suggesting that they weren't really listening to the internal signals. And when we measured their caloric intake in the lab, we found that the YFAST was a better predictor of caloric intake, and that it occurred above and beyond body mass index. So suggesting that even in these young children, what we're seeing is potentially this greater consumption of these foods, and that they are a major target for food marketing. And I'm even working with a pediatrician to try and understand how we might understand differences in reward reactivity and the impact of very early exposures in the first years of life to high levels of sugar and ultra processed foods in the development of reward systems in infants over time. Because we can't wait, really, until even an 8-year-old has had thousands and thousands of experiences and learning and advertising and modeling, that it's hard to kind of get at the sense of what is the impact early on in those earliest exposures to sugar. So, for example, one of the studies that we recently did found that infants who were given sucrose to numb pain in response to a heel stick, that infants who showed the greatest response right after they were born and when they were newborns to sucrose, that it showed more pain numbing effects, suggesting it was more effective at activating endogenous opioid response, that those babies actually had more rapid weight gain over the first 2 years of life than infants who showed less of a responsiveness to sugar and what is likely the opioid system. So it seems like we can really start to try and understand how might this be playing out from the moments of birth, even in utero, and understand how are those kind of reward profiles going to interact with dietary expansion, where babies go from milk to a more adult diet and are more likely to have an added sugar food or beverage by the time they're 2 than they are a fruit or vegetable on any given day. Like what does that do? We don't really know right now. I want to kind of finish very quickly on the idea of food advertising, because I've talked kind of a lot in this clinical realm, but I strongly believe that what we've learned from addiction is that if you have an environment that puts so much pressure on the individual to overconsume, and you're constantly targeted and cued and triggered, that being able to approach with just intervention is not going to solve a public health crisis. And one of the policy efforts that I think of that's potentially at our fingertips, if we have the political will and that other countries are doing, is restricting food advertising, at least in the context of children and adolescents. So, just quickly, we know that children are seeing a lot of food commercials. I think I just updated my review and it's now up to 10,000 food commercials every year, or about 10,000 food commercials on any given year. They're almost all for these foods that we think might be addictive, and advertising has actually increased to adolescents at 12 years of age and older. So we did some pilot work where we looked at how neural differences and brain regions that are so highly associated with addiction, like the caudate, like the nucleus accumbens, whether adolescents who showed greater activation in these brain regions to food advertising were more likely to gain weight over time. And I'll skip the commercial because you all know what a McDonald's commercial looks like. But what we did find is that adolescents who showed greater activation in the caudate at baseline to these fast food commercials relative to control commercials of phones showed the greatest weight gain over time, and that these brain regions in response to these food advertisements also predicted greater consumption specifically of unhealthy foods in my simulated fast food restaurant. I'll give you a quick glance at that guy. You can kind of see it over there on the side, and you can see our little menu, as that the above and beyond self-report, above and beyond how much they said they liked these advertisements for these junk foods, that the brain response of this greater activation in the caudate and the nucleus accumbens was a greater predictor of overall food intake in my lab, and specifically unhealthy food intake. So I'm going to pause there and go to my thank you slide, and provide us with an opportunity to take questions. Again, here's my little fast food restaurant lab, and feel free to go to my website to learn more about the studies that we're doing. All the scales that I mentioned and more that we've developed, including an expectancy scale, are freely available on my website, and you can get our publications and some other talks I've given on this topic. And now I'm happy to open it up to answer questions in our remaining time.
Great. Thank you so much, Ashley, for a very stimulating talk. That generated a bunch of questions. Let's try to get through a few of them. What's the role of demographic variables, including socioeconomic status, in your measures of food addiction?
Yes, that's an excellent question. So generally what we're seeing is that we're not necessarily seeing differences by race, ethnicity, which is a little surprising to us. I'm sorry. Oh, can you hear me okay? But because we know that individuals of color are more targeted by food advertisers for unhealthy foods, and often what we're finding is that food insecurity, in a recent study that we've recently found, is that food insecurity itself is highly associated with more addictive like eating on the Y-fast. And I think this kind of boils down to the sorts of foods that are available to individuals who have food insecurity. They're almost more often what they have available and what's cheap and subsidized are ultra processed foods that might be addictive. And then there's this intermittency where individuals kind of get a lot of access to these foods, and then by the end of the month, they kind of run out of foods. And so there's periods of deprivation. And if we know one of the behavioral systems that's most likely to trigger an addictive response and sensitize the dopamine system is intermittent access. And so I think food insecurity and social justice and racial justice issues about who has access to what types of calories is absolutely key to thinking about how do we promote health.
Lovely. What about sodas, diet sodas? What's their contribution to some of these factors?
Oh man, the artificial sweetener debate is such a hot topic. So we definitely do see that sugar sweetened beverages are heightened in the context of individuals who have this more addictive like eating profile. That isn't surprising given that you both have caffeine, which we know has psychoactive effects, plus the additional effects of sugar, and this is heightened. In a cross-sectional work that we've done in the Harvard Nurses Health Study, we do see heightened intake of artificial sweeteners and diet foods, diet sodas, diet foods that are associated as well with higher YFAS scores. And one of the things that's hard to disentangle at this point, given that this work is cross-sectional and this human model is this a reaction? Are these people who are gaining weight more rapidly and are trying to reduce their reliance on sugared food, so they've kind of switched to diet foods and artificial sweeteners? Or are the artificial sweeteners kind of contributing to metabolic changes and kind of certain preferences for intensities of sweetness that are then contributing to the development of an addictive process? And I would say that's a very open question, and one that's really important to understand, and that Dana Small's group is, I think, also moving forward by not just looking at artificial sweeteners in isolation, but thinking about that, combining them with other sources of energy intake and seeing very different profiles of how it affects metabolism in the brain based on what the artificial sweeteners are consumed with. So it's a complex question that really needs more rigorous research.
Just have time for another question or two. So the role of genetics, polymorphisms, dopamine, and also intergenerational transfer.
Yes. So I would say there hasn't been a ton of work yet on the potential genotypes that most be associated. There is a little bit of work in one study where they did find an association with a dopamine multi-locus profile and a greater likelihood of food addiction. That was done by Carolyn Davis's group. There was another study in the Harvard Nurses House study that didn't necessarily see an association with that genotype. I would say overall, the YFAS being, a relatively new tool, it hasn't gotten into the really huge representative samples with really large numbers to do very rigorous and reliable genotyping. And I also think so much that the genotype is going to interact so much with the environment. And so when it comes to food, we really need to kind of have both of those contributors in mind when trying to figure out whether it, just like you kind of see with alcohol, but there does seem to be some research but needs to be replicated in larger, more rigorous samples. The other piece is that we have seen in our early work that parents who have higher YFAS scores also have children with higher YFAS scores. The same is true with our withdrawal scale. We've created a child version of that and have found the same time course and the same correlates, and that parents who reported that they themselves had more addictive eating had children who are more likely to exhibit withdrawal when they tried to improve the dietary intake of the child. And I think of that as being really relevant when we think of interventions that are done to help reduce childhood obesity, is that we're often doing a family systems approach to try and get the whole family to improve their dietary intake. And I think, man, if a parent is going through withdrawal and having an addictive drive towards these foods, and at the same time their child is going through withdrawal and having this addictive pull towards these foods, and what we're seeing is it leads to kind of more oppositional and problematic behavior on the type for the child, and that they're harder to parent, that we really do maybe need to design our treatments in a more nuanced way to be able to support these families that are as a whole experiencing this addictive phenotype, both of them.
Thank you very much. That was a wonderful talk, and we're at the end of our time. I'd like to thank everyone for attending, and have a wonderful day.